First of all, I would like to thank Dave Allison for the invitation. I appreciate that, although he's not here. You guys can relay the message. Number two, I would like to really thank you for the hospitality. The meat was outstanding. Telling you, talking about obesity and control of food intake, forget it. Just forget it. I'll be a hypocrite, but if you have a chance, just go there and dig in. You're going to live once. You might as well live it right. So that's what I think, at least. On personal basis, you've heard that I got my DDM degree from Baghdad University. Although I'm Jordanian, by the way, I'm not Iraqi. So I just lived there until I finished my degree, and then was fortunate enough to come to the States and do my training at K-State, and then at Purdue and University of Illinois in equine surgery. Then after that, the WSU for the PhD, working with my guardian angel, Bob Ritter. If you haven't heard of him, you probably will if you want to study control of food intake. Kirk might know him, definitely. I agree, right? Yep. It's good seeing you, by the way. Thank you. Today, what I will do is I will tell you the story that I've been interested in to study the control of food intake and investigating one of the most important systems in the body, the gastrointestinal tract. And I'll tell you, I hope I'll convince you that this system is a very important system in terms of controlling our food intake. Well, first of all, because that's where food goes in and out, so might as well, be an important system. And I will present to you data today on two of the satiety peptides, cholecystokinin, or CCK, and gastrin-releasing peptide, or GRP. And I'll tell you, the first thing is I'll divide the presentation into two parts. The first part is working our way from the outside toward the inside, from the outside toward the gut. And then after that, I will show you, hopefully, convincing evidence about the role of the actual gut in the control of food intake by these two satiety peptides. So the presentation is going to be divided into two parts, if you will. Okay, so another thing about my style of presenting is that you will have couple of styles, or probably three styles. One style that you cannot ask questions until the end of the presentation. Another style is you can ask whenever you want. A third style is you get in and out without absolutely knowing what was going on. This is my third part, my presentation. But no, the presentation, I encourage you to ask any questions if you have any. Interrupt me, I could care less. Okay, but don't go to the next point without understanding the previous point, okay? At least I cannot do it. If you can, then be my guest. But this is my style. So ask whenever you like. Okay, so let's start this journey. So the outline for the presentations are going to be, I'm going to be talking about obesity. What's obesity? And I know all of you probably are familiar with this topic. A couple of facts about CCK, cholecystokinin, and gastrin-releasing peptide. Then we'll talk about the gastrointestinal tract, my favorite system. And then we'll talk about a hypothesis and show some of the data that we've generated in the lab for the past few years or so. And then we'll conclude. Okay, so I don't think I'll have any problem convincing anyone in this room or outside this room that we have a problem of obesity, not only in the Western world, but all over the place, basically, and especially in our beautiful southern states. Alabama, Mississippi, Georgia, you name it. And this is reflected, by the way, also in the prevalence of the diseases that are associated to obesity. And this is a table probably all of you have seen from the internet, from the CDC, 2013, showing that Alabama has the highest prevalence in type 2 diabetes. Therefore, I mean, this sets the stage pretty well for obesity as a disease, and in order for us to hopefully find a cure for obesity, and is to understand the disease. If you don't understand it, it's pretty hard to cure it. So that's basically what we're going to do. What we're hoping in the lab is basically, as part of the effort, if you will, all of us here, is basically to participate in the understanding of the disease in order for us, hopefully, to find a cure for it. So what is basically obesity? And again, the cliche, you've heard it, you've seen it on CNN, Fox News, everywhere under the sun, is the fact that, okay, well, it's an imbalance between energy intake and energy expenditure. Okay, so energy intake and energy expenditure. Basically, what you do is we have more intake than what we spend. And if you are a gentleman, you will end up with this extra intake in your abdominal cavity, versus if you are a female, you're going to end up having this extra fat in your butt, basically. Hips in general. Okay? So basically this is the basic thing about obesity. It's extra intake, and that intake is being stored as fat. Okay. So if this is the bottom line, so how am I going to cure it? Well, I guess the logical thing is to control food intake. Excellent. We solved the problem. So, control of food intake. And as far as we're concerned, there are a couple of components that we will be talking about. The control of food intake for a longer period of time, and we call that the long-term control of food intake, and basically it involves zillion factors: genetics, exercise, you name it, your family, your family tree, or whatever. I mean, it's a whole host of things. Okay, excellent. And then the next part is what we will call the short-term control of food intake. And the short-term control of food intake consists of two components: the size of individual meals and the time between two consecutive meals. Say breakfast and lunch, lunch and dinner, dinner and breakfast the second day, and so on and so forth. In my talk and in my research, I'm focused on the short-term control of food intake. Short-term control of food intake. Why? Because it's easy. Okay? Because it's measurable. Okay? You can determine it. Okay? I cannot determine your genetics. Okay? I'm not Steve Watts or Dave Allison. Okay? I can measure how much you can eat per meal. Pretty easy. And if you're a rat, that's even easier. You put it in a biodax system, in and out, the meal size, intermeal interval, you name it. That's what I like. Easy. Okay? And if you're a good graduate student, pick an easy problem to fix. Don't go with the complicated stuff. You'll be stupid. Now, the key behind these, let me explain to you how this works. Any creature under the sun, whether it's a zebrafish or a jellyfish or a tilapia or Ayman or a cow or a horse or whatever. This is basically how we portray our feeding behavior. We're hungry. So we eat all the way until we are full. Done. This is a meal. Now you have a period of resting, sleeping, going with your girlfriend, having sex, you name it, right? Different behaviors. Then you're hungry again. You start eating again. That's the second meal. Okay. So, we are concerned with this first meal and the time between the first and the second meal. And why are we interested in that? Very simple. Because most of the peptides that regulate these two components of the short term of food intake are basically secreted from the gastrointestinal tract. This is why at the beginning we started to become interested in the gastrointestinal tract. The four peptides that regulates meal size and the inner meal interval are basically secreted from the gastrointestinal tract. And here they are highlighted: CCK or cholecystokinin, gastrin-releasing peptide, or GRP. We have also GLP-1, and we have PYY, and other less important hormones. These are the peptides that reduces these two components, meal size and prolongs the inner meal interval. Others may prolong it, like ghrelin, right? But we're not concerned about that. So the data I'm going to show you are basically from what CCK and gastrin-releasing peptide. Okay. I'll give you a little history about them, just key facts, so you will remember them. Before we dive into the data, I know you guys are anxious to see data, right? And results and all this good stuff. But before you go to the data, you need to understand the basics behind the data, so you will understand what the data means. This is very important. Now, in the 70s and in the early 80s, a couple of peptides were discovered, cholecystokinin and GRP. 73, a group of Cornell, Jerry Smith and James Gibbs documented that CCK reduces food intake. After that, Steve Woods at Cincinnati, at that time at University of Washington, Seattle, documented that it reduces meal size and it prolongs the inner meal interval. Okay, now we know what the meal size is, we know what the inner meal interval is, right? Excellent. So, they did not mention that these two peptides come in different forms based on the amino acid number, or the number of amino acids in the peptide chain. So you have a number of forms. You have CCK8, 8 amino acids. You have CCK33, the original form, and you have CCK58, which is the only, I repeat, the only endocrine form of CCK in rats and in human. The only endocrine form of CCK in rats and in human. 58 amino acids. Keep this information in the back of your mind, so we'll come back to it a little later. Now, for GRP, we have again 3 forms, again based on the number of amino acids in the peptide and in the peptide chain, and we have GRP10, the small GRP, exists in everybody under the sun. We have GRP27, exists in everybody except the rat, and we have GRP29, which exists only in the rat. Again, keep this information for the remaining of this presentation, because the data will be presented on 2 forms of the CCK and GRP, CCK58 and GRP29. Why? Because no one used them except us. That's another thing for the graduate students to choose, right? Choose something that no one did before. So at least you're going to get your papers published, right? Or for the faculty to get tenured or to get grants. Okay. CCK works on two G protein coupled receptors, CCK1 or A and CCKB receptor. A for the alimentary tract or the GI tract, B is for the brain. Okay? And then GRP also works on three G protein coupled receptors, BB1, BB2, and BB3. The main one for GRP is BB2, highest affinity, and again, I'll show you, that's also distributed in the gut. Role of feeding, I mentioned to you the early studies of Jerry Smith and James Gibbs. Anyone can refer to them and go there. Next, I will show you the piece of data that we actually added to the literature, and that is the effect of GRP, especially at 29, because it's the only endocrine form in rats, and the rat is our beloved model to study obesity and control of food intake. Therefore, the data here will show you GRP, or the effect of GRP 29 on meal size and inner meal interval. These are spray dolly rats, fasted overnight, and the test diet is a 10% sucrose solution. On the x-axis, you will see the different doses of GRP, and on the y-axis, you will see the intake in mil. The first slide, or the upper graph represent the meal size, and the lower graph represent the length of the inner meal interval, time between meal 1 and meal 2 in minutes. And of course, we've tested 10 and 27 just to make the reviewers happy, right? Now, as you can see with GRP 10, which is basically the dotted bar or the last bar in the graphs, you can see that 4 out of the 5 doses reduced the size of the first meal, and 5 of the doses prolonged the inner meal interval. So this is basically like what Jerry and Jim did with CCK. That GRP 29 also reduces food intake, basically by reducing meal size and prolonging the inner meal interval. So this is the role that has been established. Excellent. So, okay. What is our hypothesis then? Our hypothesis is basic, very basic, that the gut contains regulating sites that controls meal size and inner meal interval length by CCK and GRP 29 and CCK 58 again. Okay? The gut has a role in the control of food intake, or the short-term control of food intake, by CCK 58 and GRP 29. And the reason for that, why did we choose the gut? We chose it for several reasons. And I'm going to go over these reasons to convince you that the gut is actually worth investigating and worthy of our time and effort, right? Excellent. So, everything starts with the histology of the gastrointestinal tract, okay? And here, this picture shows the different layers of the gastrointestinal tract. This is the luminal side, this is the inside of the gut, the lumen, and this is the outside of the gut, or we call it the serosal surface, okay? If you're a surgeon, you don't care about this surface. Why? Because it's not a holding layer. You don't care for it. When you put your needle in it, it goes through it. You don't care for it. The main thing that you care for is this layer here that we call the submucosa. And I know you're probably not interested in surgery, but you will be pretty soon. Now, back to the layers of the gastrointestinal tract. The first layer is the mucosa, which consists of 3 layers, epithelial layers, lamina propria and muscularis and mucosae, then the submucosa, then 2 muscle layers, inner circular and outer longitudinal, and then the final layer which is the serosal layer. Now, sandwiched between the two muscle layers, the blue layer right here, we call that the myenteric plexus. And under the submucosal layer, we have what we call the submucosal plexus. Collectively, myenteric and submucosal plexus form what we will call later the enteric nervous system, or the second brain, or the brain of the gut. Okay? Very important. So if you cut all the innervation to the gastrointestinal tract, the outside innervation, the gut still functions. How? By the enteric nervous system, the myenteric and the submucosal plexuses. Now, why this is important? This is important because the enteric neurons are the source of GRP, gastrin releasing peptide. That's why the gut is important, right? Number one. Number two, these neurons, as well as the muscle layers of the gastrointestinal tract, contains GRP receptors. Number one. Number two, the gastrointestinal tract is connected neurally or neuronally to the central nervous system by two main nerves. The nerve that you all are familiar with is the vagus nerve, okay? And that goes and ends up all the way in the DVC, or the dorsal vagal complex. Area postrema, nucleus tractus solitarius, and dorsal motor nucleus of the vagus. DMV, not DVM, okay? So NTS, DMV, and AP, or area postrema. That's where the feeding, or the central feeding centers, at least in the hindbrain, are located. The other nerve that connects the GI tract or the gastrointestinal tract to the central nervous system, we call it the splanchnic nerve. The splanchnic nerves ends all the way in the dorsal root ganglia on your spinal cord or beside your spinal cord, and goes all the way to reach also the DVC, which means that it does have a role in the control of food intake. Two nerves. Now, why am I mentioning this? Because these nerves contain receptors for both CCK and GRP as well. Okay? And these central areas also contains receptors for GRP and CCK. And here's some data that shows you the distribution of these receptors in the different gut areas. This is the esophagus, this is the stomach, this is the duodenum, then the jejunum all the way to the end of it to the ileum. These are all small intestine. And then you go to the large intestine, which is cecum, and then colon and rectum. So 3 small intestines, 3 large intestines, duodenum, jejunum, ileum, that's small, cecum, colon, and rectum, that's large. And as you can see, GRP receptor or CCK receptor distribution is all over the place, mainly in the jejunum and in the distal duodenum. DD stands for distal duodenum, and J is for jejunum. And for the GRP, most of it is basically in the fundus and in the antrum, part of the stomach. These are the antrum, and this is the fundus, okay? And most of your GRP receptors are distributed there. That is why the GI tract is very important in this hypothesis. One, because it secretes CCK and GRP. Second, the nerves that connects to the central satiety areas, at least in the hindbrain, also contain CCK receptors and GRP receptors. Number 3, GRP and CCK basically are secreted in response to food intake. So you eat high fat and there's CCK release. You eat a number of diets, carbohydrates and fat, and GRP is released as well. Therefore, the GI tract is important in this hypothesis. It should have a role in the control of food intake by CCK and GRP. And as I mentioned in the beginning, we will test this hypothesis by measuring the components of the short-term control of food intake, meal size and inner meal interval, first from the outside toward the gut, and then we'll go straight to the gut, right? And we'll see. If what I'm saying is correct, that means what? That means I should see changes in the meal size and in the inner meal interval length by these peptides. If I'm wrong, then by all means my hypothesis does not stand. Okay? All the way from the outside to the inside, and we've done a number of surgeries to test that. This is basically some data to show you, again, the upper graph is meal size, and the lower one is the inner meal interval, A and B, respectively. And we have tested about, 6 doses of GRP, all the way from 4.1 nanomole per kg, all the way to 17.2 nanomole per kg. And again, these are food-deprived rats. Meal is 10% sucrose. Why do we use 10% sucrose? Just like the same way we use the filet mignon last night at the restaurant. Because it's good, hedonic, and we eat until we blow in the face. As simple as that. So, okay. What we've done is we've done 4 surgeries. We've done a vagotomy, pretty simple, silly procedure. The vagus nerve in both branches run alongside your esophagus. You pick it up, cut it, pick it up, cut it, vagotomy is done. You let the animals recover and you give them CCK 58 or GRP 29, and what? You measure meal size and inner meal interval. Very simple, right? The other one is a little different. What we do is we take the source of the sympathetic innervations, the splanchnic nerve. How do we do that? We do that very simply by a microvascular surgical procedure, and that is by taking out the sympathetic ganglia, the celiaco-mesenteric ganglia. It's located between the celiac artery and the cranial mesenteric artery, 2 and 3. These are the main major branches coming off the aorta. Aorta is the artery that supplies you, your whole body, with blood. Okay? So the major branches, 2 and 3, are celiac artery and what? Cranial mesenteric artery. So between them, in the picture before, you will see this unclear, hasty, fibrous connective tissue-like material. Believe it or not, this is not a fibrous connective tissue material. This is nerve cells, and this is the sympathetic ganglia. And you take it with forceps and scissors. So this is number 1, and here it is after the what? After the surgery, right? This is sympathectomy, or we call it celiaco-mesenteric ganglionectomy. You know, if you want to impress somebody in a meeting, celiaco-mesenteric ganglionectomy, right? It's sympathectomy. Okay, so back to the data. Again, let me just point out that the end or the last graph, if you will, it's called myo or myotomy. We'll talk about that later. Okay, let's leave it now. You will see the same slide, but again later. Again, here we measured food intake, and in the vagotomy you can see there is no difference in the meal size, whereas when we took the sympathetic ganglia, that's given GRP 29, the effect is gone. So that means, hey, there is a need for this sympathetic ganglia in the reduction of meal size by GRP 29. Whereas in the prolongation of the inner meal interval, we can see in the sham there was 3 out of the 5 or of the 6 doses prolonged the inner meal interval relative to saline control, whereas in vagotomy there's none, sympathectomy there's none, and combined sympathectomy and vagotomy there's none. That means the two nerves are important for the prolongation of the inner meal interval, whereas in meal size probably the sympathetic is more important. Again, we'll talk about the myotomy later on. Now, what we did is we wanted to, again, convince ourselves as well as the reviewers that what we're talking about actually holds. And we've done FTOS immunohistochemistry. FTOS-like immunoreactivity basically is a marker for neuronal activation. And this is a cross-section from the hindbrain, the areas that control food intake. And this is the area postrema, AP, NTS, nucleus tractus solitarius. Again, in meetings, you want to impress somebody, you speak in Latin, right? So they can think that you know what you're talking about. The DMV, or the dorsal motor nucleus of the vagus, right? That's where cell bodies for the vagus nerves are located. And in A, you can see these three regions are lit like a Christmas tree, right? With these black dots. These black dots are FTOS-positive immunoreactive neurons. That's in response to CCK58. That means CCK58 was able to activate the areas that control food intake in the what? In the hindbrain. Okay, excellent job. Now, the gut. This is the hypothesis. What you see here is basically a peel, just like you peel an orange. We peel the layers of the gastrointestinal tract one by one, and we keep the enteric neurons. This is part of the myenteric plexus, and you can see also in A that all the neurons are lit up like a Christmas tree, right? That means they are FTOS positive. That means CCK58 was able to activate the enteric neurons as well as the neurons of the dorsal vagal complex, the areas that control food intake. And you have to believe me that the same picture, we saw it in the submucosal plexus. You have to trust me on that, okay? Because we said that the enteric nervous system consists of how many plexuses? Two plexuses. Myenteric plexus and what? A submucosal plexus. Myenteric sandwiched between the muscles, and submucosal plexus laying underneath the submucosal layers. The submucosal layer is very important if you're a surgeon. Why? Because it holds your needle. Why? Because it's strong and it's fibrous connective tissue. And I'll show you why in a second when I show you the myotomy procedure that I told you. We'll talk about it in a little bit. Okay? Excellent. So, CCK activates the DVC and the enteric neurons. We've demonstrated this activation also by what? By the use or the utilization of the CCK1 receptor antagonist, davazepide. We've tested this activation, and as you can see in the middle picture, FTOS is gone when you have a davazepide pre-treated animal. So CCK1 receptor or A receptor was necessary for the activation of the enteric neurons by CCK58. Whereas in C, CCK2 receptor antagonist or L365-260 did not do a thing. Why? Because we still have FTOS immunoreactive neurons in the myenteric plexus. Excellent. So activation took place, reduction of food intake took place. The activation and the reduction were basically CCK1 or CCKA receptor mediated. Now the next one is to see what is the deal with the nerves. Cut the nerves and see if this activation is dependent on the vagus or not. Okay, well, this picture or these photomicrographs, 4 of them, the upper ones, A and B, represents the dorsal vagal complex, DVC, and the lower ones represent the myenteric plexus. In the right-hand side, which is B, it's vagotomized. When we cut the vagus, you can see that all the activation in the dorsal vagal complex is basically gone. Whereas when we cut the vagus in D, the enteric neurons activation is what? Continues to be there. That means it's vagally what? independent. So cut the vagus or not, who cares, right? That's what we show here. And we showed you in the food intake data that when we cut the vagus, remember the 4 surgeries? Nothing happened. That's different than the usual CCK8 story where you cut the vagus and subdiaphragmatic vagotomy or deafferentation and all this good stuff, you eliminate the reduction. Well, guess what? When you use the right form of CCK, which is CCK58, which is the only endocrine form of CCK in the rat and in humans, you get different results. How about that? Now, we went further to see if the enteric neurons, which you have about 100 million neurons in the enteric nervous system, all of which contain different neurotransmitters, different shapes, different functions, then how are you going to say that, okay, CCK activates this population of enteric neurons versus this, okay? We have a number of techniques to do that. And one of them is based on the morphology. Based on the morphology. This work has been done in our lab. You can see these pictures. They have different shapes. For example, in A, we have a larger cell with long dendrites. We call these Dogiel type 2 neurons. Okay? Alexander Dogiel is a Russian histologist who actually characterized these neurons, right? And they're called by his name. Okay, so this is one, Dogiel type 2. Whereas in C, for example, smaller, shorter dendrites, if you will, and multiple dendrites. So here you see one long one in A, here you see 3 different ones in C, and these are Dogiel type 1. When you know the function of these neurons by doing what we call chemical coding. Chemical coding is to know the structure of the or the neurotransmitter in these neurons, you will find out that CCK activates, this is double labeling now, FTOS and something else. You will see that, that CCK activated mostly NOS immunoreactive neurons. NOS is a marker for inhibitory motor neurons. Meaning that if you activate these neurons, what's going to happen to the gut movement or motility? Inhibited. These are inhibitory motor neurons, right? Remember that, okay? Others, they are excitatory motor neurons. When they're excited, they increase motility. You go to the bathroom more. Diarrhea, we call that in medicine, right? Versus when you activate these guys, what do we call that? Constipation, right? So CCK basically activates these neurons. That is why CCK is involved in what? In inhibition of what? Of gastric emptying as well, right? Because it inhibits the movement of the gut. It inhibits the movement of the stomach, and therefore the food stays in the stomach for a longer period of time. Excellent. So now we're in the periphery, we're in the vagus, sympathetic, etc. Here we will take you, or I will take you, if you allow me, deeper into the gut wall. And what I will do is I will take you through another microsurgery, and that is myotomy. And by myotomy I mean cutting through the layers of the what? Of the gastrointestinal wall. Except what? Which layer we should preserve? God, you guys are sleepy. Or there's hypoglycemia in the audience. Which layer is important here? A surgeon. The submucosal layer, because it's the holding layer, because it's the fibrous connective tissue layer, because if you open that layer, the gut contents are going to be out, right? Peritonitis, death. Well, guess what? These are living, breathing animals that you do food intake on, right? So they're alive. Why? Because the myotomy procedure cuts the layer and the nerves above the submucosa. It does not touch the what? The mucosa. The submucosa, I'm sorry. Okay, and then we do the food intake, as I said. And here is the data. This is the previous graph that I showed you before. And you can see that 3 out of the 6 doses, at least the 3 lower doses, basically were eliminated in myotomy. That means the enteric neurons are important. or are, not, I don't want to say required, but they're important. They have a role, right? They have a role. And again, all of the doses basically were not effective in the myotomized animals in the prolongation of the intermeal interval, as I can see in this thing. Once the mouse move, I can tell you. Yeah, here. Okay? So that means the enteric neurons, the gut, is important in this effect. Now, all of this will take us basically to the technique that we are using now, and we call intra-arterial catheterization. I showed you the aorta before when I did the sympathectomy, if you remember. And this is the first branch, which is the celiac artery, and this is the second branch, which is the cranial mesenteric artery. Now, I will show you a microvascular procedure that we do, that we catheterize these arteries in rats. Rats are breathing, living animals, and we do food intake on these rats. So instead of doing the cannula in the brain, I do it in the gastrointestinal tract. Why did I choose this technique? I choose this technique because it tests the specific area of the gut that is supplied by each of these arteries or their branches. So if I'm talking about the celiac artery, I'm talking about stomach and upper duodenum. If I'm talking about the cranial mesenteric artery, I'm talking about small and part of the large intestine. The three is the caudal mesenteric artery, which we did not catheterize. Why? Because it supplies the rectum, and we don't care for the rectum at this point. Right? Okay, so let me describe to you the surgery pretty quickly. Again, in A, it's the normal anatomy. 1 is the aorta, 2 is the celiac, 3 is the cranial mesenteric artery. In B, we ligate at the base of the cranial mesenteric artery. In C, we puncture the cranial mesenteric artery with the tip of a 25 gauge needle. This is a microscopic surgery. Don't try this at home. And then in D, we insert the catheter in the artery, and then we secure it with super glue. We then test if what we did is correct, and we inject a dye that's called latex. And we can see that with the celiac injection, we only label the stomach and upper duodenum, which is pretty correct. And then with the cranial mesenteric artery, we label small and part of the large intestine. Once the animals are recovered, we go and we test food intake, meal size and intermeal interval. And this animal are basically, or these experiments are done in biodac system, freely fed rats, ad libitum. You eat as much as you want, just like going to Texas de Brasilia. Okay? And we gave a number of doses of CCK58. Also, I want you to remember that the doses used here with this technique is probably hundreds of times less than used intraperitoneally and by other routes. And we can see that the celiac artery is basically having most of the fun, action for reduction of meal size, whereas a couple of the doses for the CMA. Meaning what? Meaning that not only the gut is responsible for regulating meal size and intermeal interval by CCK58. The same data, by the way, shows for the GRP29, but for the sake of time, I'm showing just synopsis of each one of them. Not only that the gut controls meal size and intermeal interval, but the most important thing that we've discovered after these experiments is the fact that the sites, or the gastrointestinal tract sites that control these feeding responses are different. It's not buy one, get one free. Not everything controls meal size and food intake and stuff like that. No. Specific sites are responsible for controlling food intake, for controlling meal size, versus other sites in the gut controlling what? Intermeal interval. This is what the data shows. The CA was responsible for the meal size reduction, celiac artery, stomach and upper duodenum only. As you can see, CMA, only the highest dose. We don't care for it. But when we switch to the next one, we saw the effect more in the CMA for the intermeal interval. That's very important. Now we want to talk to Kirk's buddy, Randy Seeley, and we tell him, okay, well, Rand, does these peptide have the same effect in a gastric bypass animal? And if they do, would they reduce it to the same level as the regular rat, or would they use it or reduce it more? We've done the surgeries based on collaboration with Randy Seeley, now at Michigan, I believe, Michigan, right? Yeah, and then we've modified the technique. I like modifying things. I don't like to stay with the status quo. So, you know, cut and throw it to the dogs, basically. So we cut piece of the stomach, and then we anastomosed B with B and A with A. The orange with the orange, and this is the Y arm of the gastric roux and Y. That's where the Y came from. And here a simple end-to-end anastomosis, and God bless America. Surgery is done, animals are recovered, and we measured meal size and inner meal interval in these rats. Okay? Excellent. We measured it. Remember that these rats have what? Have the catheters in the arteries. So they have dual surgeries. They're not only having gastric bypass, but also they have catheters in the celiac artery, cranial mesenteric artery, a femoral artery as a control, and we have it in the portal vein. I cut the portal vein because there was no effect. You have to trust me on that. If not, just go to the paper, PubMed, and you'll see it. Okay, so again, GRP 29, most of the action, 5 doses, 4 out of the 5 doses reduced meal size, again in the celiac artery. Guess what? The reduction in the gastric bypass was more for the meal size. Now, look at the inner meal interval difference. Look at the A in the sham versus B in the gastric bypass guys. Way more. We have the same y-axis, by the way. Okay? So we're not trying to fool you or anything, right? And look at the satiety ratio. The amount of food consumed per a unit of time. We call that the satiety ratio. When you divide the inner meal interval by the meal size, you call that satiety ratio. The amount of food consumed per a unit of time. Look how much they ate per unit of time. Way, way more than the sham guys, right? So these peptides are really, 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 really functional in these animals. We have, this is preliminary, we have more reduction in cumulative food intake as well. And finally, when we tried to do this, we tried to see that, okay, if these are the effects, would they reduce the body weight of these rats? And we did a preliminary study like this for about 14 days. We give them daily injections, in this case GRP 29, and we have seen in diet-induced obese rats, these guys are maintained on 62% fat. So we're not talking McDonald's here. We're talking really fat. I mean, good stuff, right? I'm telling you. And we found that the guys, the prone and the resistant of these guys, they were reducing in body weight. This is basically just a preliminary study, so I don't want to draw too many conclusions out of this, and I don't want you to get too excited about it. Okay? So the most important thing is there is reduction in body weight. Okay? And maybe the excitement will come next year when I come here next year and talk to you more. Okay, so in conclusions, we will say that I've shown data to support the hypothesis that the gut has a role in the control of the short-term control of food intake by CCK and GRP. I hope you agree with me on that, and I hope I did convince you with that. Number one. And number two, this is very important for us, is that there are different sites in the gut that regulate the different feeding responses by these two peptides at least. Now, in our future work, what we will do is basically we will try different cocktails of these peptides to see their effects on body weight and meal size and inner meal interval. Hopefully we'll find something very interesting. Some of the papers that are published recently, they've tried, CCK agonists and also GLPs, GLP-1s, I should say, and they found phenomenal results. So we're hoping that the future work will lead us, or we are going in that direction. At the end, again, thank you for the invitation. The people who did the work, Martha Washington and a bunch of graduate students, interns, etc. Our collaborators, Reeve is UCLA Medical School. He's the one who discovered CCK58 and GRP29. Gibbs and Jerry Smith from Cornell, they're the godfathers of the field of control of food intake. Randy and Berger are the ones who taught us how to do the gastric bypass. Tim Moran at Hopkins, who've been with us all the way, of course, on these things. And of course, funding by NIH and other sources, both veterinary and human drug companies. Thank you. I hope you liked it.